Okay, just think about the world around you for a second. Inside, outside, even, inside us. It's almost overwhelming when you consider the microbial world. It's so incredibly vast, just teeming with life. Millions, maybe trillions of species. And the really wild part, most of them we haven't even grown in a lab, let alone described. So the big question is, how do we even start to figure out who's actually living there? And maybe more importantly, what are they doing?
Exactly. That's the core challenge. And it's why this toolkit has evolved.
Right, and that's what we're diving into today, this incredible toolkit that microbial ecologists use. It's like getting a backstage pass to the detective work? Uncovering all these microbial secrets.
Yeah, our source material today really lays it all out. It's a chapter just packed with methods. We're talking everything from like the really old school techniques, enrichment cultures and stuff, right up to the absolute cutting edge. Molecular tools, single cell analysis. It's amazing stuff.
So the mission today is to unpack these tools, understand how they work.
Pretty much. How they work, and crucially, what they've actually shown us about microbes in nature, the kind of surprises they've turned up.
Okay, so get ready. We're going from like scraping your tongue to diving to the bottom of the ocean, all through this lens of microbial analysis. Let's unpack this. So for the longest time, I mean decades really, if you wanted to understand a microbe, step one was always grow it.
Right. Yes, absolutely. And, culturing is still fundamental. It's really the only way to fully pin down all its properties, its metabolism, how fast it grows, who it interacts with, all that detail.
You need that pure culture.
To really define its niche and predict its impact. Yeah, you ideally need it isolated in the lab.
Okay, so one of the classic ways to get towards that, especially from a messy environmental sample, is the enrichment culture. How does that work?
Right. It's basically about manipulating the conditions. You set up a specific environment in the lab, the nutrients, temperature, oxygen, whatever, to really favor the growth of the organism you're looking for.
While discouraging everything else.
Exactly. You're trying to mimic its preferred niche, but maybe turbocharge it a bit to help it outcompete the others in the flask.
So, like, if I wanted to find those nitrogen-fixing bacteria, azotobacter, from soil.
You set up a clever trap. You use a liquid medium, but with no fixed nitrogen source, no ammonia, no nitrate, but plenty of carbon. Incubate it in air.
Okay, so anything that can't fix its own nitrogen just can't grow.
Right. And anything that needs anaerobic conditions won't grow either because of the air. So you're strongly selecting for aerobic nitrogen fixers. Azotobacter often thrives in those conditions.
Makes sense. And what about the Winogradsky column? That always looks so cool. It seems like a whole ecosystem enrichment.
Oh, it's a beautiful demonstration, yeah. It's like building a layered microbial world in a bottle. You pack mud, organics, sulfate, add water, shine light on it.
And different layers develop over time.
Exactly. Based on these gradients, yeah. Oxygen from the top, sulfide bubbling up from the bottom. So you get sulfate reducers deep down, then different photosynthetic bacteria in colored bands where light and sulfide meet, aerobes at the very top. It's a great source for isolating different functional types.
But there's a catch with these enrichments, isn't there? Especially the liquid ones, something called enrichment biases.
Yes, and it's a big one. A significant limitation. The problem is, the conditions you create, often with lots of nutrients, might just favor the microbes that grow the absolute fastest in the lab.
Not necessarily the most important ones out in nature.
Precisely. They might not be the most abundant or ecologically relevant in the original spot, where conditions are usually much tougher. You can end up selecting for these lab weeds, basically. Diluting the initial sample can help a bit, but the bias is always a risk.
Okay, so getting a positive enrichment means the organism was there, maybe even just one cell started it off. But it doesn't really tell you how important it was in the grand scheme of things.
Exactly. Proof of presence, yes. Ecological significance. Not so much from the enrichment alone. And importantly, if you fail to enrich something, it doesn't mean it's absent.
Right. Your conditions might just have been wrong.
Totally. You can never prove a negative with enrichments.
Okay, so let's say you do get something growing. You still need a pure culture, right? Just one type of microbe. What are the standard ways to get that isolation?
Yeah, the classic methods involve dilution and solid media, usually agar plates. The streak plate is probably the most common, right? You dilute the culture and spread it out so individual cells grow into isolated colonies.
Each colony from one original cell, ideally.
Ideally, yeah. Then there are agar dilution tubes, which are good for anaerobes. You mix the sample into molten agar, let it solidify, and colonies grow embedded inside. And liquid dilution, like for the most probable number or MPN counts, can sometimes get you down to a single cell type, starting a culture in a tube. but it's less common for isolation itself.
And then you have to be really sure it is pure.
Oh, absolutely. That's critical. You check colony shapes, look under the microscope to see if all the cells look the same, maybe test growth in different media.
See if anything else pops up.
Exactly. And nowadays, sequencing a key gene like the 16S RNA gene from the isolate is pretty much standard practice to confirm purity.
Okay, that's the classic workflow. But what about those microbes that just refuse to play ball, the really low abundance ones, or the super slow growers that get lost in the mix?
Right, that's where we need to get more targeted. Single-cell isolation methods are key here. Things like laser tweezers. It's amazing.
Laser tweezers. What's
Okay.
that?
Use a focused laser beam, like tiny optical fingers under a microscope, to physically grab a single cell you're interested in and just pull it away from everything else.
Wow, like microscopic fishing.
It's a perfect analogy, yeah.
Right.
And then there's flow cytometry, which is much faster, much higher throughput.
How does that work?
It shoots cells, single file, through a laser beam. And it measures how they scatter light or if they fluoresce naturally or with a stain. Based on those signals, it can physically sort cells into different tubes, thousands per second.
So you can separate specific types from a huge mix?
Exactly.
Right.
Very quickly. You can sort populations based on size or maybe fluorescence if you've tagged them somehow.
And people have actually used this sorting to finally culture some of those difficult microbes.
Yes. High-throughput culture methods often use flow cytometry. They sort individual cells, or very small numbers of cells, into thousands of tiny wells on microtiter plates. Each well is like a mini culture.
Testing tons of conditions at once.
Exactly. You can try hundreds or thousands of slightly different media recipes simultaneously, often adding things like sterile seawater or soil extract, hoping to provide some missing nutrient or growth factor. This was absolutely key for finally getting Pelagibacter ubique into culture.
The super abundant ocean microbe.
Yeah, one of the most abundant bacteria on the planet.
Yep.
It just wouldn't grow using standard methods because it's adapted to such low nutrient levels. High throughput, dilution to extinction culturing finally cracked it.
Okay, so culturing is powerful for digging deep into one organism, but we know it misses a huge chunk of the community. So how do we study microbes in their environment without the culture step?
Right, this is where the culture-independent revolution happened.
Right.
And believe it or not, the microscope is still a cornerstone, just a much more sophisticated microscope now.
What can you see just using basic stains on a natural sample, like mud or water?
Well, general fluorescent stains like DAPI or SYBR green, they bind to DNA. So under the right light, all the cells glow because they all have DNA. It lets you see and count the total number of microbial cells there. Some stains can even give you a rough idea of live versus dead cells.
And can you actually tag specific cells to watch them?
Yes, with fluorescent proteins like GFP, green fluorescent protein. Now, you can't just add GFP to wild microbes.
All right, they don't make it naturally.
Exactly.
Yeah.
But you can take a microbe, you can culture, engineer it to produce GFP, and then reintroduce it into an environment. The fluorescence lets you track where it goes, how it survives. You can even link GFP expression to specific genes to see when and where those genes are turned on.
But the big frustration with just looking, right? You see all these tiny dots and rods. They all look the same, but you know they're different species.
Oh, absolutely. Morphology is incredibly misleading in the microbial world. So many different species, even whole phyla, can look identical under the microscope. The source has a great figure showing this looks like one type of cell by normal light, but specific stains reveal multiple distinct lineages right next to each other.
So how do you see that hidden phylogenetic diversity? How do you know who is who?
That's where FISH comes in, fluorescence in situ hybridization. It's a game changer.
Okay, FISH. It uses fluorescent probes, right? Little bits of DNA or RNA.
Yep, synthetic nucleic acid probes with a fluorescent dye attached. And these probes are designed to stick or hybridize, specifically to complementary sequences inside the microbial cells.
So you design them to target sequences unique to certain groups.
Exactly. For identifying microbes, the target is almost always ribosomal RNA. And that's clever for two reasons. One, cells are packed with ribosomes, so there's lots of RNA, giving a strong signal.
Makes the whole cell light up.
Right. And two, RNA genes have regions that change very slowly, useful for broad probes targeting all bacteria, for example, but also regions that change much faster, allowing you to design probes specific to a genus or even a single species.
So you can paint different microbes, different colors right there in the sample.
Pretty much, you can take a slice of soil or a biofilm, treat it with differently colored fish probes, and literally see which bacteria are next to which archaea or where your specific target species is living, and roughly how many there are compared to the total DPI-stained cells. Identity, location, abundance, all visualized.
But I remember reading there was a catch: slow-growing cells might not have enough ribosomes to light up well.
That's true. Low ribosome content can be an issue for standard phage sensitivity, making faint cells hard to see, which spurred development of amplification methods. But FISH fundamentally answers the "who is there" question. The next big question is always, okay, but what are they doing? Can you see activity with these methods too?
Yeah, can you actually visualize their metabolism somehow?
You can. Specialized techniques combine the identification power of FISH with ways to measure metabolic activity, again at the single cell level. Things like cardfish and BONCAT-FISH are really powerful here.
Okay, cardfish, BONCAT-FISH. How do they show activity?
They target different aspects of the cell's activity. Cardfish often targets messenger RNA, the transcripts, indicating a gene is actively being expressed, and it uses an enzymatic amplification step to really boost the fluorescent signal, which helps with those low ribosome cells. BONCAT-FISH measures translation, whether the cell is actively making new proteins.
Making proteins, like, are they growing or producing enzymes right now?
Exactly. BONCAT uses a neat trick. You feed the sample a modified amino acid, say, a version of methionine with a little chemical handle attached.
Okay.
Active cells, busy making proteins, will incorporate this tagged amino acid. Then you add a fluorescent dye that specifically clicks onto that handle.
So only the cells that were building proteins light up with that second color.
Precisely. And if you combine that with a phylogenetic FIF probe of a different color, you can simultaneously see which specific microbes, identified by FISH, were actively translating proteins lit up by BONCAT when you took the sample. The source has a great image showing this in a marsh. Some FISH-identified cells are bright with BONCAT, others aren't. It links identity and activity beautifully.
That's incredibly cool, seeing who's busy and who's not. Okay, so microscopy with these tags is powerful, but still, you're looking at potentially billions of cells. Analyzing that manually seems daunting. Which brings us to DNA sequencing, right? That really blew the doors open.
Oh, absolutely. DNA sequencing, especially the high-throughput methods, completely changed microbial ecology. Suddenly, we could get massive amounts of genetic information directly from the environment. No culturing needed, initially anyway.
And PCR is the workhorse there, making copies of specific genes.
Yeah, polymerase chain reaction. You extract the total DNA from your sample soil, water, whatever, which contains DNA from potentially thousands of species. Then you use PCR primers designed to amplify a specific gene you're interested in.
Like the 16S rRNA gene, the sort of universal barcode.
That's the classic target for phylogeny, yeah. Because everyone has it, but the sequence varies enough to tell different microbes apart. Or you could target functional genes, genes involved in nitrogen fixation, sulfate reduction, methane production. Table 19.3 lists a ton of common functional gene targets.
So you amplify that one target gene from everyone in the sample who has it, ending up with this big pool of slightly different versions of the gene.
That's the first step, exactly, like in figure 19.16. The big challenge, especially early on, was how to sort out that mix. How do you figure out how many different versions, phylotypes are there, and how abundant each one is?
How did they do that before the modern sequencing machines?
They used clever but ultimately limited fingerprinting techniques. Things like DGGE, denaturing gradient gel electrophoresis, or TRFLP, terminal restriction fragment length polymorphism.
Right, I remember seeing those gel pictures with all the bands.
Yeah, DGGE separated DNA fragments based on sequence differences affecting how they melt, creating a band pattern. TRFLP used restriction enzymes to cut the fragments, and the length of the end piece varied by sequence. ERISA was another one, looking at length variation between RNA genes. These methods gave you a snapshot mainly of the most abundant types in your sample.
But they couldn't really resolve all the minor players or tell apart very similar sequences.
Exactly. The resolution was limited, and they definitely underestimated the true diversity, which is why next-generation sequencing, or high-throughput sequencing, was such a paradigm shift.
It seems like going from a magnifying glass to the Hubble telescope almost.
That's not a bad analogy. Instead of looking at bands on a gel or cloning and sequencing things one by one, these machines could sequence millions or even billions of DNA fragments all at the same time, directly from that PCR pool, figure 19.19.
And that sheer volume of data just revealed a whole hidden layer of diversity.
Totally. Suddenly you could see the rare biosphere all those phylotypes present at really low abundance that were completely invisible before. And these rare members might be doing important things or could become dominant if conditions change. Early studies using this tech, like Craig Venter's Sargasso-Sea project, immediately found tons of new microbial groups and genes missed by all previous methods.
Okay, so if sequencing the 16S gene tells you who is there, then metagenomics takes it further, sequencing all the DNA, right, to see all the
Yeah.
genes.
Yes, that's the idea behind metagenomics. Sequence everything, get a collective blueprint of the entire community's genetic potential. The big challenge then becomes computational, taking those billions of short sequence reads and trying to assemble them back into genes and ideally link those genes back to the genomes of the organisms they came from.
Which is hard in really complex samples like soil.
Extremely hard in soil, yeah,
Yeah.
figure 19.23. But it's getting better. We can often assemble partial genomes or even near-complete metagenome assembled genomes or MAGs, especially from less complex environments like some marine waters, figure 19.22. This helps link function genes to phylogeny.
What were some of those early mind-blowing finds for metagenomics, like that Sargasso Sea work?
Oh, huge discoveries came out of that, even though they only scratched the surface. They found genes for ammonia oxidation that clearly belonged to archaea. Nobody expected that.
Yeah.
It led directly to identifying the Thamarchiota, which we now know are critical ammonia oxidizers globally. And they found proteorgopsin genes, allowing bacteria to use light energy in lineages where they were totally unexpected. It fundamentally changed our view of energy capture in the ocean.
But even with all that power, metagenomics is still just showing the potential, right? The blueprint, not the actual activity.
Exactly. It tells you what genes are present, what the community could potentially do. It doesn't tell you if those genes are turned on, how fast reactions are happening, what the microbes prefer to eat, or how they respond to stress. And discovering truly novel functions just from sequence is still really tough, which loops back to why culturing remains so important to actually test the biology.
Okay, so metagenomics is the whole genetic blueprint. Is there a way to get a more focused look at the functional potential without sequencing everything?
Yes, microarrays can do that. In environmental work, they're often called geochips. These are like little slides dotted with thousands of probes, but instead of targeting just anything, these probes are specifically designed to bind to known genes involved in key metabolic processes: carbon cycling, nitrogen cycling, sulfur cycling, and so on, figure 19.20, table 19.3.
So you take the total DNA from your sample.
Label it, usually with a fluorescent dye, and wash it over the chip. The DNA fragments will stick to the spots containing probes for the genes they match.
And the brighter the spot, the more of that gene was in your sample.
Pretty much. The intensity of the fluorescence on each spot gives you a relative idea of the abundance of different functional genes. It's a quick way to survey the potential metabolic capabilities encoded in the community's DNA.
But again, potential, not actual activity.
Correct. Same limitation as metagenomics in that sense. It tells you the toolbox is there based on the genes, but not which tools are currently being used. And there can be technical issues with cross-hybridization, similar genes sometimes sticking to the wrong probe.
Right. So to really get at what the community is doing, right now you need to look beyond the DNA. You need more omics.
Exactly. This is where the idea of environmental multi-omics comes in. You integrate data from different levels, genes, transcripts, proteins, metabolites, to build a much more complete picture of function, figure 19.21.
So connecting the metagenome, the potential, with something like the metatranscriptome, what's that?
Metatranscriptomics means sequencing all the messenger RNA, mRNA, in the sample. Since mRNA is the intermediate step between a gene, DNA, and a protein, looking at mRNA tells you which genes are actively being transcribed, being expressed, by the community at the time of sampling.
So it's a snapshot of active gene expression.
Yes. It gives you a much better idea of which metabolic pathways are actually switched on. Figure 19.25 shows an example related to nutrient cycling in coastal waters.
Okay, and metaproteomics goes even further, looking at the proteins themselves.
Right. Proteins are the actual enzymes, the transporters, the structural bits. They're the molecules doing the work. So metaproteomics analyzes all the proteins present in the community, usually using mass spectrometry to identify them.
And ideally, you link those proteins back to the genes and organisms.
Ideally, yes.
Yeah.
Having a metagenome from the same sample is really helpful for that, matching the detected proteins back to the predicted genes and the organisms they came from, like the permafrost methane metabolism study in figure 19.26. It gives you a direct look at the functional machinery.
And the metabolomics, that looks at the small molecules.
Exactly. Metabolomics analyzes all the small molecule metabolites, the inputs, outputs, and intermediates of metabolism. Things like sugars, amino acids, organic acids, including molecules released outside the cells, the exometabolites. This arguably gives you the most direct view of the community's food web.
Like, who's eating what and who's producing waste products that someone else might use?
Precisely. That study on biological soil crusts is a fantastic example, figure 19.27. Metabolomics showed how the main cyanobacterium was releasing a whole cocktail of compounds that filled the other microbes living with it, and it suggested those other microbes might be giving nitrogen compounds back. It revealed this intricate metabolic exchange network.
So layering all these omics, genome, transcriptome, proteome, metabolome lets you connect the who to the what they're doing and even the how they're interacting.
That's the goal, yeah. It provides this incredibly rich systems-level view. It helps understand how diversity translates into function and how communities might respond to change, like predicting carbon release from warming permafrost based on the combined omic signals.
Okay. Besides looking at molecules like RNA and proteins, are there more direct ways to measure microbial activity in a sample? Maybe simpler ways?
Sure, you can do straightforward chemical measurements. If you know a specific reaction occurs, like sulfate reducers consuming lactate and producing sulfide, you can just measure the concentration changes of lactate and sulfide over time in an incubation. Figure 19.29a.
Simple, if the signal is strong enough.
Exactly. But if you need more sensitivity, or you want to track where atoms are going, or measure rates very precisely, then isotopes are the way to go, especially radioactive isotopes initially.
Right, using tracers, like adding radioactive CO2, 14 CO2 to measure photosynthesis rate.
Perfect example,
Yeah.
figure 19.29b. Or adding radioactive sulfate, 35SO4, to measure sulfate reduction, figure 19.29c. Or maybe 14C labeled glucose to measure how fast it's respired to 14CO2. Figure 19.29d. You add the labeled food, incubate, and then measure how much radioactivity ends up in the product or in the microbes themselves.
But how do you make sure it's the microbes doing it and not just some random chemical reaction?
Controls.
Yeah.
Absolutely essential. The key one is the killed control. You run a parallel sample where you've killed the microbes, usually with a chemical like formalin or maybe by autoclaving it. If the reaction stops or drastically slows down to the killed sample, you know it was biological.
Makes sense. You always need that baseline.
Yep, and controls without the added labeled substrate too to check for background noise.
Are there other chemical tricks, maybe inhibitors, to block certain pathways?
Yes, inhibitors can be useful. Acetylene's a classic one. It messes with enzymes in nitrification and denitrification. But it's probably most famous for the acetylene reduction assay.
For nitrogen fixation, how does that work again?
Nitrogenase, the enzyme that fixes N2 gas, is unusual. It can also reduce molecules with a triple bond. Acetylene has a triple bond, so you give the sample acetylene instead of N2 gas, and the nitrogenase reduces it to ethylene.
And ethylene is easy to measure.
Much easier and more sensitive than measuring tiny changes in N2 gas. And since very few other things reduce acetylene like that, it's a pretty reliable proxy for nitrogenase activity. Figure 19.3.
Okay, that's clever. What about getting really fine-scale measurements, like right inside a biofilm or down into sediment layers?
For that, you need microsensors. These are amazing, incredibly fine glass needles. The tips can be just a few micrometers wide, and they're designed to measure specific chemicals: oxygen, pH, nitrate, sulfide.
So you can poke them into a sample and see how things change over tiny distances?
Exactly. Use a precise micromanipulator to move the sensor step by step, maybe just 50 or 100 micrometers at a time, building up a detailed profile. Figure 19.32 shows classic profiles in sediment. You see oxygen disappear rapidly just below the surface, while nitrate might penetrate deeper.
You can map the chemical microenvironment.
Precisely. You can even use arrays of different microsensors together. They've even put them on deep-sea robotic landers to measure processes right on the ocean floor. Figure 19.33.
Wow. And nanosensors are even smaller for complex surfaces.
Yeah, these are tiny particles, often fluorescent or phosphorescent. You can spray them onto irregular surfaces, like a coral or a biofilm. The light they emit changes based on the chemical concentration right next to them, like oxygen.
So you can visualize gradients on bumpy things.
Right. The source shows a great example using them on coral, figure 19.34. You could see high oxygen patches where the symbiotic algae were photosynthesizing in the light, and then oxygen-depleted zones in the dark where the coral itself was respiring. Really cool spatial information.
Okay, we talked radioactive isotopes for tracing activity, but you also mentioned stable isotopes, the non-radioactive ones. How do they fit in?
Right. Stable isotopes. Think carbon-12 and carbon-13, or sulfur-32 and sulfur-34. They're chemically almost identical, but enzymes often have a slight preference, usually for the lighter one.
They discriminate a little bit.
Exactly.
Yeah.
This is called isotopic fractionation, figure 19.35. The result is that the biological product often ends up enriched in the lighter isotope compared to the starting material. It leaves a kind of isotopic fingerprint.
And this fingerprint tells you something about the process.
It can, yeah. For instance, the carbon in plants, and thus fossil fuels, is isotopically lighter than the CO2 they started with, because photosynthesis enzymes prefer 12C, figure 19.36. Methane from methanogens is even lighter. These signatures in ancient rocks are actually some of the key evidence for early life on Earth. Same with sulfur. Sulfate reducers strongly prefer 32S, leaving a distinct signature in sulfide minerals, figure 19.37.
So it tells you if biology was involved, maybe way back when, but can you use stable isotopes to see who's doing what now?
Yes, that's the really exciting application. Stable isotope probing, or SIP. This is how you link activity directly to identity in a complex community today.
How does SAP work?
You feed the community a substrate, maybe glucose, maybe ammonia, maybe even N2 gas that's been artificially enriched with a heavy stable isotope. So 13C glucose instead of normal 12C glucose, or 15N ammonium instead of 14N ammonium.
Okay, you feed them the heavy food.
Right. Now, only the organisms that are actively metabolizing and incorporating that specific substrate will build that heavy isotope into their own biomass, into their proteins, their lipids, and crucially, into their DNA.
So their DNA becomes slightly heavier.
Exactly. It's physically denser than the DNA from all the inactive microbes or those eating something else. And you can exploit that density difference.
How do you separate it out?
Using density gradient centrifugation, figure 19.38. You spin the total extracted DNA really fast in a special salt solution that forms a density gradient. The heavy DNA containing the 13C or 15N sinks just a little bit further down the tube than the normal light DNA.
And you collect that heavy fraction.
Yep. Got that fraction, and now you have DNA primarily from the organisms that were actively consuming your labeled substrate. Then you can sequence a do 16S PCR to see who they are, maybe sequence the whole metagenome from that fraction to see their full capabilities.
Wow, that is super clever, directly linking the meal to the diner. Okay, so many of these techniques, sequencing pools of DNA, measuring bulk gas changes, they look at the community level. But what about getting down to the nitty-gritty? What's a single cell doing? Especially one you can't culture. That seems like the real frontier.
It absolutely is. Linking genetics and activity right down to individual, uncultured cells is where a lot of the cutting-edge work is happening.
And NanoSims is one tool for that. What does it do?
NanoSims is secondary ion mass spectrometry, but at the nanoscale, figure 19.39. It uses a very fine ion beam to basically sandblast the surface of a single cell. It knocks off ions from the cell.
And measures what comes off?
Yes. A mass spectrometer analyzes those ions, telling you the elemental and isotopic composition of that tiny spot on that single cell. So if you did a stable isotope feeding experiment first, say with 15N.
NanoSims can tell you which individual cells actually took up the 15N.
Exactly. You can map the isotope enrichment within a single cell. And if you combine it with FISH beforehand to identify the cell, you've directly linked identity and activity at the ultimate resolution.
Okay. And MAR-FISH. Sounds similar to BONCAT-FISH, but with radioactivity.
Similar goal. Yeah, linking identity and activity visually. MAR-FISH combines microautoradiography, MAR, with FISH, figure 19.41. Here you feed the sample a radioactively labeled substrate, maybe 14CO2 or a tritiated 3H compound.
So active cells become radioactive.
Right. And you do FISH to label the cells you're interested in. Then you coat the whole thing with photographic emulsion, like old school film. The radiation emitted by the active cells exposes the emulsion right around them, creating tiny black silver grains.
So you look for your fluorescent FISH labeled cell and see if it has silver grains clustered around it.
Precisely. Silver grains mean it took up the radioactive food. No grains, no uptake. Again, identity linked to activity. You can even estimate activity level by counting the grains. The main limitation is having the right radioisotope available for your process.
And Raman microspectroscopy, another single-cell technique.
Yes, and this one's cool because it's non-destructive. You can use it on live cells.
Yeah.
Figure 19.40. You shine a laser on a single cell, and the molecules inside scatter that light in a characteristic way. You get a whole spectrum, like a chemical fingerprint of the cell.
And isotopes change the fingerprint.
They do. If you feed cells a stable isotope like 13C, the peaks in the Raman spectrum associated with carbon bonds will shift slightly, so you can see which cells incorporated the label. It can also sometimes identify specific compounds or tell you about the cell's physiological state.
Okay. These single-cell microscopy methods are incredible, but finding and analyzing enough individual cells in a complex sample, that still sounds like finding needles in haystacks.
It can be. Which brings us back to flow cytometry, figure 19.42, but now used in a much more integrated way. Remember, it can analyze thousands of cells per second.
Right, and sort them.
Exactly. So now you can combine flow cytometry with things like fish staining, or maybe the BONCAT activity stain. You can tell the cytometer. Sort me all the cells that are positive for this fish probe and also positive for the BONCAT signal.
So you can physically isolate just the active members of a specific group.
Precisely. Or you can even use it for index sorting, where it records the fluorescence properties of a single cell as it sorts it into a specific well of a plate. Then you analyze that single cell further.
Like that early example of sorting out Prochlorococcus and Synechococcus in the ocean and realizing how many there were.
That was a landmark use case, yeah. Showing the power of sorting based on intrinsic fluorescence and size. Now we can sort based on specific probes and activity markers.
And once you sorted that single, maybe uncultured cell into a well,
Yeah.
can you get its genome from just one cell?
Yes. This is single-cell genomics, and it's truly revolutionary. The key technology is something called multiple displacement amplification, or ADA, figure 19.43.
MDA, what does that do?
It takes the tiny, tiny amount of DNA from that single cell and makes millions or billions of copies of the entire genome, more or less randomly. It provides enough DNA material to then feed into a next-generation sequencer.
Without needing specific PCR primers that might miss things.
Right. MDA aims for whole genome amplification, using random primers, trying to avoid the bias you might get if you only use PCR with specific primers.
So, let me get this straight. You can take a scoop of seawater, use flow cytometry to pick out a single uncultured bacterial cell based on a FISH probe, use MDA to copy its entire genome, and then sequence that genome to figure out what it might be capable of doing.
That's the power right there. That's the workflow that's unlocking the secrets of the so-called microbial dark matter, the vast majority of life we couldn't study before because we couldn't culture it. And sometimes the genomic information gives you just the clue you need to finally design the right culture medium to grow it after all.
Wow. Okay, that has been an absolutely incredible tour through this toolkit. I mean, from simple broth in a flask and streaking plates to, shooting lasers at single cells, sorting them in streams of fluid, sequencing billions of DNA bits using tiny sensors. It's amazing.
It really shows the creativity involved, doesn't it?
Uh-huh.
All these methods, often used together now, are what allow microbial ecologists to tackle those huge questions. Who's there and what are they doing?
In all these different places, our bodies, soil, oceans, sediments, even like the pipes in our house.
Everywhere.
Right.
And they're constantly revealing surprising things, like you said.
Yeah, like the structured communities on our tongues shown by FISH, or that hidden ocean diversity from sequencing, the complex food webs and soil crusts from multi-omics, the potential climate feedbacks from permafrost microbes.
Exactly. We're getting this increasingly detailed picture of microbial life and just how critical it is for, well, everything. Ecosystem function, nutrient cycles. We've moved so far beyond thinking of microbes as just single species in a test tube.
It definitely feels like the better the tools get, the more complexity we find, not less. It doesn't seem to be getting simpler.
That's absolutely true. Even with all this power, the sequencing depth, the single-cell tools, we still can't fully characterize everything in just a gram of soil or a milliliter of seawater.
Yeah.
It's just too diverse. That rare biosphere we can now detect, that alone represents this huge unknown frontier.
Right. So even though these tools have shown us so much, the microbial world clearly has tons of secrets left to uncover. It really makes you wonder, doesn't it? What crucial jobs might those rare microbes, or the ones we still can't culture, be doing out there that we don't even have a clue about yet?